Genetic manipulation of animal cells. So previously we have focused at how animal cell lines they can be transformed by using different transfection methods. In addition to animal cells, now we also have got the ability to synthesize transgenic animals, where we are not going only to study the function or expression of genes into the cells, rather at the level of whole animal. तो इसका मतलब है कि हमने कुछ ऐसी टेक्निक्स डेवलप कर ली हैं जिसकी मदद से हम जर्म लाइन को भी ट्रांसफेक्ट करवा सकते हैं. जर्म लाइन्स ऐसे सेल्स हैं जो बाद में गैमीट्स फॉर्मेशन करते हैं. तो वंस अगर जर्म्स लाइन ट्रांसफेक्ट हो जाएं तो वो करेक्टर जनरेशन टू जनरेशन चलता रहता है. तो इस तरह से हम कुछ ऐसे बायो एसिड्स भी कर सकते हैं जो कि ना कि सेलुलर लेवल पे हों, रादर एट होल एनिमल लेवल. सो दिस इज द टॉपिक दैट वी आर गोइंग टू फोकस. In addition to animal cells, we are shifting our focus onto whole animal. That how certain genes they can be expressed at animal level. Now, if we focus on animals rather than plant cells, then we will have the idea that plant cells they have the ability of regeneration. That after transformation, certain cells of the plants they can be transformed into callus, that is undifferentiated or disorganized mass of cells, and then they again can be regenerated into whole plants. But animal cells mostly they lack this ability, that they cannot be mostly regenerated into whole animals, like if we use the somatic cells. Here there may be different reasons. पहली रीज़न तो ये है कि इसमें जो somatic cells हैं उसमें like the plant cells जो totipotency है, that is ability to de-differentiate, वो lack करती है. कि एनिमल सेल्स में ये एबिलिटी मोस्टली नहीं होती कि वो सोमैटिक सेल्स उसके रीजनरेट कर जाएं. और दूसरा, अगर सोमैटिक सेल्स जो हैं वो ट्रांसफॉर्म हो भी जाएं तो ये जो ट्रांसफॉर्मेशन है ये में भी ट्रांजेंट हो, टेम्पोरेरी हो, अंटिल के जर्म लाइन में जो ट्रांसफेक्शन जो है वो ना हो. वंस जर्म लाइन्स दे हैव बीन ट्रांसफॉर्म्ड, देन we can regenerate whole animals. So here again we are going to focus on some methods for the production of transgenic mice, that how we can develop transgenic animals like mice from the germ lines. And it is one of the most important discovery of the 20th century that now we have the ability to generate or synthesize in the laboratory the whole transgenic animals. And in case of transgenic animals, we are not only going to evaluate the expression or function of the genes, but such animals they can be used as a bioreactors for the synthesis of some vital proteins, for example in the milk. But here not all cell lines from the animal they can be used for transfection. Some specific cell types we can consider are taken into account, like the oocyte. Oocyte, it is a cell in the ovary that is after meiotic division going to develop into an ovum. We can consider such type of cells for transformation, and even some embryonic stem cells that can be commonly called as ES, or embryonic stem cells, or ES cells. So such cells like oocyte or embryonic cells from the mother, they can be taken and foreign DNA, it can be inserted. And briefly, it can be grown in vitro, and then it can be reintroduced into a foster mother. Foster mother का मतलब है कि ओरिजिनल मदर नहीं है, जैसा कि केयरिंग टाइप अगर किसी का एटीट्यूड हो, कोई और किसी का कोई चाइल्ड लेके उसका ब्रॉटअप किया जाए, तो उस टाइप ऑफ मदर को फॉस्टर मदर कहते हैं कि एक मदर से सेल लिया, दूसरे ने उसकी परवरिश की. तो इस तरह से फॉस्टर मदर को हम यूज करके तो होल जो एनिमल है उसको जनरेट कर सकते हैं इन केस ऑफ माइस. Now again we will here focus that what are the different techniques for the generation of transgenic mice. So here we are going to discuss the summary. If we consider from the sperm that DNA can be transfected into the sperm and then it can be used to fertilized, unfertilized mouse egg here. Another method is that even after fertilization has taken place, we can inject our gene of interest by using some physical methods like DNA microinjection. So here we can transfect even fertilized egg. Once egg fertilized and foreign DNA has been inserted, then it can enter into further cell division and develop a structure very common that is called as the blastocyst, where hollow structure it is formed. And ultimately it can lead into embryonic development. So DNA can be introduced into the sperm that can result in the fertilization or even within the fertilized egg. In addition to that, DNA, which is exogenous in origin, it can be used for the transfection of adult cells or even for the ES or the embryonic stem cells. So this technique can be used that either adult cells, somatic cells can be transfected or embryonic cells. And another method can be used that the recombinant retroviral infection can be carried out for the infection of the embryonic stem cells. So ultimately, all this genomic DNA, which is exogenous in origin, it can be used for the transfection of either fertilized egg cells or early embryonic cells. So ultimately, embryonic development is there. So these are different techniques or methods that can be used for the generation of transgenic mice.